Hi, I'm Nicholas Walker, Vice President of Data Science and Informatics at Enhanc3D Genomics, and it's a pleasure to introduce you to the company and explain our mission to transform disease treatments through our unique platform for three-dimensional profiling of genomes. As you know, our body is made up of trillions of cells that perform specialized functions such as neurons in the brain or immune cells for fighting infection. Remarkably, most of these cells pack nearly 2 meters of DNA into their nucleus in an intricately folded structure. What makes this more remarkable is that the intricate structure determines the function of the cell. For example, whether the cell is a brain cell, a liver cell, or a heart cell. And it also determines the state of the cell, such as whether, in the case of an immune cell, it's ready to fight infection, or it's abnormal, which could lead to disease. Enhanc3D Genomics is a technology platform that allows us to measure in one shot the 3D interactions of 22,000 genes. The platform operates down to 50,000 cells, making this tractable for individual tissue biopsy. And it is backed by a powerful data science and bioinformatics suite for interpretation. The platform can be powerfully applied in many areas, but I want to explain one really important application. With the advent of sequencing, thousands of individuals have had their genome sequenced, and areas of the genome have been linked to disease, represented here by a Manhattan plot, which is a typical output of a genome-wide association study. And surprisingly, what scientists have found is that most of the regions associated with disease are not local to genes, which code for RNA and produce proteins that make the cell function, but they turn out to be distal to genes and difficult to link to genes. Only that is how it seems with normal sequencing. In fact, after 3D genome sequencing, they can be mapped properly to the genes that they impact. I hope it's intuitively clear how 3D genomics can be used to identify gene targets for disease treatment. But identifying targets for disease treatment is one thing, but Enhanc3D Genomics can go a step further than that. By measuring the DNA interaction, we link the control site of the gene to the gene. Thus, the control site can be developed as a biomarker for the treatment target. And that provides a route to profile individuals which the treatment would most effectively work in. And this is a foundation to precision and personalized medicine. So let me recap for a moment. Enhanc3D Genomics is the first technology platform that can screen 3D genomes for 22,000 genes efficiently. The technology is able to work with a low amount of material, making it practical to profile individual tissue biopsies. Now, those 2 points together enable Enhanc3D Genomics to cost-effectively and comprehensively profile healthy and diseased 3D genome maps, enabling a foundation for new disease treatments and precision medicine approaches. This in turn feeds to our data science program and clinical network to generate novel therapeutic development strategies and IP assets. And this can also lead to a high-value pharma services business model. We've recently announced our platform, GeneLinked, and at the same time announced being awarded the Johnson & Johnson Quickfire Challenge for the application of this technology platform in immune-mediated disease, validating our approach by one of the world's leading pharmaceutical companies. With these announcements, we're now actively advancing programs in autoimmune disease, aging, and immunology. I want to give you an idea of how we're building value in what we call our value cycle. We believe our technology can transform the development of therapeutics by using human data. And that gives a much higher chance for successful clinical application. Our approach generates hypotheses from patient-centered data sets, and then we employ our platform to measure 3D functional states comprehensively in patients and across relevant tissue. And then our data science platform allows us to interpret and funnel those core target predictions to functional validation. And then the outputs of that are once again fed back into our hypothesis building loop. So we're on quite a journey here. The pink circles show our expected growth trajectory over our therapeutic development program. And we further expect, with the maturation of our technology platform, it will see new approaches in several other market arenas, such as cell engineering and high-value reinterpretation of the millions of linear sequenced genomes that are accumulating. We have a deeply qualified team that has a passion to see the impact of this technology on disease, and we'd love to hear from you if you want to join this opportunity to enable precision medicine. Thank you very much for your time.